All right, this is chapter 43, part 3, humoral and cell-mediated. So humoral is B cells, which involve the secretion of antibodies as they become plasma cells. And cell-mediated response incorporates T cells, mainly, specifically cytotoxic T cells or killer T cells. So here you see everything sort of together, humoral and cell-mediated, and how they incorporate or cross the two. Helper T cells, the conductor here, communicate with both B cells and cytotoxic T after they've been relayed the information from antigen-presenting cells. Specifically here in this case, this is dendritic cells using MHC2. And then the B cells will give rise to memory B and also the plasma cells, which will secrete the antibodies. And the cytotoxic T will give rise to memory T and active T, and then you also have memory helper T. Now, helper T cells produce a protein called CD4 that helps them to bind to the antigen-presenting cells, specifically the class II MHC molecules. And I describe it as kind of like a grappling hook. It activates the helper T, and then when it docks, and I'll show you a picture here in a second, when they dock together, when the CD4 binds with the MHC2 and draws the cells together, cytokines are released, signaling kind of like we've docked, continue the process, and it helps to stimulate other cells. Those cytokines help to stimulate other lymphocytes. So here you see, for example, our helper T cell, and here's a dendritic cell, consumes a pathogen. This is an antigen-presenting cell. MHC2 takes these antigens, displays it on the surface. Here you see the CD4 surface protein that binds to the MHC2 and draws the two together. And when it binds, that signals sort of docking has occurred. And cytokines are released from the helper T cell that triggers or stimulates this specific cytotoxic T cells and specific B cells for that antigen that recognize excuse me, that recognize that antigen. All right. And then there's also CD8. So helper T cells are CD4, and cytotoxic T cells make CD8 protein. A similar idea. But again, this time it enhances contact between cytotoxic T and the target cell. Where did I go? Remember that cytotoxic T cells work on infected cells and also cancerous cells, things that need to be defeated that are of self. Through MHC1, we can see here the MHC1 class molecule from a nucleated cell of the body. Let's say there's a viral particle inside. Here we have an antigen that's displayed. CD8 from the cytotoxic T attaches, and in response to the CD8 attaching, perforin gets released. Perforin is a protein that punches holes in the membrane of your cell, this cancerous cell, for example, and allows various toxins and things to enter, and also causes the cell to pop just simple by causing an influx of fluids because of the very hypertonic environment here. It triggers apoptosis, and here you see this cell having been popped or killed. Now, B cells, part of the humoral response, are through cytokines, as you saw here. Once again, here's cytokines, here's B cells. So now we're going to look at B cells, this specific component, through the transmission of the information through the helper T cells. And the B cells mature and they become plasma cells. And those will be the ones that secrete the antibodies. You also have a small subset of the cloned B cells that become memory cells. And here you see that taking place. Here, CD4 binding to MHC2, helper T cell communicates with B cell through cytokines and showing the antigen. And then this B cell then specifically will clone itself, release lots of antibodies that bind to the specific pathogen. And you also have some memory cells there. Now there's 5 major classes of antibodies, or immunoglobulins, that differ in function. Okay? IgM, IgG, IgA, IgE, and IgD. It's probably a good idea to be familiar with the main ideas of these immunoglobulins, and I'll go through some of them right now for you here. IgM is the first one that is produced after exposure. First class that's produced after initial exposure to an antigen. Okay, that's IgM. I don't know how you remember it. M comes first. Okay. IgG. The one thing that you can remember about IgG is that this is the one that crosses the placental barrier and confers passive immunity onto the fetus. It allows the fetus and a pregnant mother to have some immunity because the fetus does not have an immune system while in the belly. So IgG, mom gives to the baby. IgA, mom also gives to the baby in breast milk, and it also is present in various secretions. So breast milk is actually a secretion, much like sweat. So IgA is found in these secretions, and mom gives IgA to the baby. In fact, it's been shown that mothers who breastfeed produce children with stronger immune systems, in part because of the donation of this IgA to the baby. So here you see presence in breast milk confers passive immunity on a nursing infant. IgE is widely known and associated with allergic responses because IgE triggers histamine release on mast cells. IgE actually binds to mast cells and causes mast cells to release histamine. The last one, IgD, what do you want to know about it? It's largely tied to B cells. I don't think of all of them, D is probably the least important for you to know. How about let's say that? Okay, how do antibodies work? And we're going to actually stop on this slide briefly so I can start here because this is super important for you to understand.